We have a pretty short presentation today on the MNS blood group system. This is only intended to be a basic overview highlighting the important stuff for your board exam, as well as a little bit of history, which you'll come across on the next slide. The MNS system is one of the oldest known blood group systems, discovered only 26 years or so after the ABO system by the very same Karl Landsteiner, along with Labine. They were just kind of injecting human red blood cells into rabbits and kind of seeing what types of new antibody specificities would develop. Using the family data from the human blood donors, they were able to determine that the existence of these two, the M and N antigens, were antithetical. And then about 20 years later, after the advent of the antiglobulin test, Walsh and Montgomery discovered the big S antigen, which they determined was genetically linked to MNN. It took another 4 years or so before the antithetical antigen little s was discovered. And then a couple years later, the U antigen, which stood for universal, although we now know that it's not quite universal. The U antigen is absent only on red blood cells that are also big S negative and little s negative. Familial studies and later molecular genetics showed that there was a close linkage between genes controlling the expression of the MNS antigens. There's a disequilibrium in the expression of big S and little s with MNN because the genes responsible for their expression are close together on chromosome 4, meaning that any crossing over that occurs will most likely take the entire haplotype rather than just one of the alleles. This is a concept known as linkage disequilibrium. The antigen frequencies among Caucasian donors, which we'll discuss further on the next slide, occur as haplotypes as shown here, with big N and little s being the most common, and big N and big S being the least common. The big S antigen is inherited twice as often with the M antigen as it is with the N antigen, while the N antigen is inherited 5 times as often with the little s antigen as it is with the big S antigen. This chart only breaks down the phenotype frequencies without respect to those haplotypes. There isn't much difference in the M and N frequencies between Caucasian donors and donors of African descent. Roughly half are heterozygous, while a quarter are homozygous for M, and the final quarter are homozygous for N. The big S frequencies vary quite a bit, however. More than half of the Caucasian donors express the big S antigen, while less than a third of African descendants do. Therefore, far fewer African descendants are going to be little s negative. And that said, only donors of African descent will lack the U antigen. MNS antigens exhibit dosage effect, which means that antibody reactivity is substantially greater with homozygous expression than it is with heterozygous expression. The M and N antigens are located on glycophorin A, which is a glycoprotein rich in sialic acid. Glycophorin A is similar to glycophorin B, which houses the big S and little s antigens. They're nearly homologous, which means they share a lot of the same genetic data. All MNS antigens are very well developed at birth, and a hallmark of the MNN antigens, or at least antigen serology, is that they are easily destroyed by proteolytic enzymes such as physin and papain, but they're not affected at all by dithiothreatol. The big S and little s antigens, however, are located closer to the cell membrane on their glycoprotein than the M and N antigens, making them less accessible. Because of this, they're not as easily destroyed by physin treatment. And then this last line is really just a little bit of trivia for you. There's an association between glycophorin A and protein band 3, which affects the expression of WRB antigen, which is part of the Diego system. And there's an association between glycophorin B with Rh protein, which is evidenced by a significantly reduced expression of big S and little s on Rh null red blood cells. Anti-M antibodies are often naturally occurring, meaning that alloimmunization to transfuse red cells or pregnancies is not necessary for the development. Anti-M and anti-N antibodies react best at room temperature, but despite being predominantly IgM antibodies, they don't bind complement very well. For this reason, they're not considered to be clinically significant unless the reactivity persists at 37 degrees Celsius. Anti-M and N antibodies are not known to cause HDFN. They're not known to cause decreased transfused red cell survival, and they're not known to cause hemolytic transfusion reactions, mostly because they can't bind complement, and they're predominantly IgM, so they're not going to cross the placenta. Anti-big S and little s, however, are often IgG. They do bind complement, and they can cause HDFN, and they've been implicated in several instances of severe hemolytic transfusion reactions, often paired with hemoglobinuria. And then finally, we have our null phenotypes. So first, we have the U negative phenotype. These represent a partial or complete deletion of the GYPB gene, the glycophorin B, the gene that creates the glycophorin B glycoprotein, including parts that code for big S, little s, and U. These patients can develop anti-U if transfused with anyone else's blood, so if anyone that's not U negative, and anti-U has been shown to cause severe hemolytic transfusion reactions as well as HDFN. Some examples of anti-U react with apparent U negative red blood cells very weakly, and those red blood cells are said to be U variant. They have an altered glycophorin B presentation that does not express big S or little s. And next we have the ENA negative phenotype, which represents a deletion of the glycophorin A, the GYPA gene, which codes for the glycophorin A glycoprotein. These individuals are M negative and N negative with decreased sialic acid on their red blood cells. There is no true ENA antigen. It doesn't exist. It's just a term that we use to describe all other antigens present on glycophorin A glycoprotein. So for this reason, there are a few different serological presentations of anti-ENA. Some examples don't react with vicin-treated or trypsin-treated red blood cells, and some examples do, depending on the exact specificity and what's cleaved by these enzymes. Anti-ENA, because of this kind of variability, can be very difficult to differentiate from antibodies like anti-WRB, which I mentioned earlier, is linked to that glycophorin A, as well as anti-PR. And then finally, we have the MK phenotype, which was named by Metaxas and Metaxas-Buehler in 1964 for the silent GYPA and GYPB gene. So they're missing both, the whole thing. These individuals will be negative for the M, N, big S, little s, U, ENA, WRA, and WRB antigens. This phenotype is a true null of the MNS blood group system. And that brings us to our final slide. These are just the things that you have to know for your exam. So first, you want to know roughly the prevalence of each antigen in the MNS system. Both M and N are present in about 75% of donors, irrespective of ethnicity. The big S antigen is present in about 55% of Caucasians, and only about 30% of African descendants, while little s is expressed in about 89% of Caucasians and about 97% of African descendants. You definitely need to know that MNS antigens exhibit dosage. That's a kind of a main facet of them. Remember that this means that homozygous inheritance will display a greater strength of reactivity than heterozygous inheritance. You also need to know that anti-M and anti-N are predominantly IgM, so they react best at immediate spin, and that they're destroyed by ficin, as well as other proteolytic enzymes. Since they don't bind complement, they're not considered to be clinically significant unless they react at least 37 degrees Celsius. Likewise, you need to know that anti-big S and anti-little s are predominantly IgG antibodies, reacting best at IAT. Reactivity with Fison-treated cells is variable. Remember, those antigens are closer to the cell surface, so they're harder to cleave off with those proteolytic enzymes. So it's not very reliable. These antibodies are clinically significant because they do bind complement and they can produce severe hemolytic transfusion reactions as well as HDFN. And then what we just went over, remember you have the MNS null phenotypes. You've got the U negative, which is a GYPB, glycopyrin B deletion, while ENA is a glycopyrin A deletion or GYPA deletion. And then you have the MK phenotype, and that's a complete deletion of GYPA and GYPB. That's it for this lecture. Make sure you listen to the KEL blood group lecture as well. I think these two are issued simultaneously as part of the same module.